Now this next one we talk about transport proteins. This is very famous protein, right? This is very important, right? So everybody needs this protein, right? So what is the protein involved in this transport? Oxygen transport? Hemoglobin, right? So here hemoglobin is present in red blood cells, right, which efficiently carries oxygen from the lungs to the tissues of the body, right. Then hemoglobin also helps in the transportation of carbon dioxide and hydrogen ions back to the lungs. So this is the protein, right. Hemoglobin has how many subunits? 4 subunits, 2 alpha subunits and 2 beta subunits, and the B subunits they contain a specific residue called the glutamic acid, right. So this is structure of the hemoglobin B chain, it looks like this. If there is a change in the nucleotide or a change in the amino acid, so this will affect this transport, right. For example, here if you see this GAG, right. So GAG code for glutamic acid, if it is converted to GTG, right. So A is changed to T, right, then it changed glutamic acid to valine. So it changed to glutamic acid to valine. So now just now we discussed about different characteristic features. Which type of amino acid is glutamic acid? Charge, negative charge, right. So but valine? Hydrophobic, right. So if you mutate glutamic acid to valine, what will happen? You change, right. So now they form the hydrophobic environment. So because due to hydrophobic environment, so this amino acid residue valine tries to interact with the other hydrophobic residues in the other chain, right, in their hydrophobic packet. Because of this interactions, so it is not able to have the proper interaction, so it tend to form fibrils, right. Because of these fibrils, right, so they change the shape like the regular one into sickle. So now this is a regular shape, and if you make the sickle cell, now this will block this transport of this oxygen. So this is the one form the sickle cell anemia. This is the disease mainly we found to many people in Africa, right. So now this protein is very important for the carrier of the oxygen, right. Now there are other proteins called defense proteins. So what are called defense proteins? Antibodies. So because we defend ourselves, because we have to be safe from the environment, right, from the bacteria or virus or all anything. So like immunoglobulins, right. So this can recognize and precipitate or neutralize the invading bacteria or virus from the other species. Then there is another protein called thrombin, right. This thrombin is also very important because it is a blood clotting protein, right, because when you got injured, so this should be clotted, right. So this is the one which helps when we get injured, so to prevent the loss of blood, right, in the vascular system, right. Then there are some proteins, they are acting as regulatory proteins, right, which regulate the cellular and physiological activity. For example, insulin, right, this is not a very well known example. So it regulates the sugar metabolism. So if you are having diabetes, so right, you need to have insulin, right. So this is the one which regulates the cellular physiological activity. And a lot of other proteins, they are having different functions. One example is a monolin, is a protein which is also used to be a sweetener, right, it is safe. But then also other proteins like nutrient proteins, right, this is the one usually people know. If you talk about proteins, right, immediately they will tell about what are proteins means, they will tell about the you can eat beans, right. So you will get lot of proteins, right. So there are various nutrient proteins like ovalbumin that is available in egg, right, like the egg white and the casein in milk and so on. So there are various proteins, they have different functions in living systems, and we have these proteins for the proper function of our body, right, and all the cellular processes. Then if you have different types of structural important proteins, right, for this is called contractile proteins or the motile proteins, right. So for example, this actin or the myosin, they function the contractile system of the skeletal muscle, right. Then structural proteins, right, which gives the strength and protection. For example, keratin, right, and the fibrous protein, also the collagen, right. For example, you can find this in hairs and fingernails, right, and the feathers, right. So you can see these proteins, and these proteins they have some specific combination of amino acid residues, like they have the combination of this hydroxyproline, glycine and proline. So about this the higher contribution of this protein. So you can see the one third this is about the glycine, and hydroxyproline as well as the proline. So, this gives the elongated shape to this structural protein like the collagen or other protein, keratin and so on. Fine. Then first we discussed about globular proteins and the fibrous proteins, and the next step is the membrane proteins, right. As we discussed earlier, membrane proteins are embedded into membranes. So, if you see I give two types of proteins. Okay, here this is the membrane, right. So, I have the membrane surface here, this is the membrane, and these are the proteins which are embedded into membrane. So, this is the outside extracellular space, and here this is the membrane, right. And these proteins are, as we discussed earlier, two types of proteins, here this is alpha and this is beta, right. You can see this is a beta barrel conformation. So, these proteins also perform various functions like the transporters or receptors or channels and so on, right. Just I will explain with this with few examples, right. So, here is one example for the transporters, right. What is a transporter generally? Which transports the molecules, right. So, you can see the transporter is a membrane protein, right, which is involved in the movement of ions or small molecules or macromolecules, right, such as another protein also, right, across the biological membrane. So, this transports, right, mainly these transport, right, assist, right, the substance, right, mainly by the active transport, right, from the lower concentration to the higher concentration. And for example, multi-drug efflux transporter, here is an example I give for the multi-drug efflux transport ACRB, right. Here how the transport happens, okay, here you can see the molecules, right. So, here this is the inner membrane, right, here this is the periplasm and this is the outer membrane. So, here this is the protein involved in outer membrane is toll C, right, this toll C, and here the inner membrane this is the ACRB, right. So, if you have this substance here, right, the substrate is here, right, you can see the substrate, right. So, you cross the membrane and during this transport, these transmembrane helices like transmembrane helix 10, right, these are the residues which are involved in the electrostatic interactions like the ASP 407 and lyse 940 and ASP 408, right. They are disturbed, right, these are the ion pairs are possible candidates for the translocation, they are disturbed. So, the helix transmembrane helix 10 would change the conformation and goes here, and due to the pumping mechanism this will be pulled out through the outer membrane to the outside. So, this is how they do the transport mechanism. Likewise, there are various transporters which are known in the literature, but they have different mechanisms to transport ions and the molecules across the membrane. Proteins, right, these are also membrane proteins, for example, ion channels, right. There are different types of ion channels, right, potassium dependent ion channel, chlorine dependent ion channel and so on, and these proteins also they cross the membrane, right, selectively they will cross the membrane. There are two properties for this transport. So, one is the selective ion conduction and the second one is gating. So, one is selective ions means this will select only some ions, okay, for example, they select potassium but not chlorine, right. Likewise, they select some sort of selectivity, so this will allow only to some ions. And the second one is the gating. Gating means for some cases it will open the channel, so it will allow to pass through, and some cases it will close the channel. So, in this case nobody can, the molecules they cannot enter into these channels, right. Okay, so, if you see this example, so this is the example for the closed and open conformation. So, here if you see the molecule is here, the ion is here, they want to transport through this membrane, right, and here if you see this is glutamic acid 148, this 148 is here inside the membrane. So, in this case it blocks the transport, right. Now, it change the conformation and here the E148 moves away from the membrane to outside. Now, the path is open and now this ion can pass through this membrane, right. This is the in, this out. So, here is blocked and here this you have the space, so it can go, right. So, there are two different aspects. One is the selective ion conduction, they will allow to pass only specific ions, and the second one is the gating. So, it has open and closed conformation, right, change the conformation to allow to pass through into the membrane, right. Okay, this is another example called olfactory receptors, right. So, olfaction is one of the senses involved in the perception of chemo signals, right. So, how to get these olfactions? Taste and smell, right. So, if you see the malaria, so it is one of the major causes because the mosquitoes can recognize human, right. This is why mosquitoes bite and then cause this malaria. If mosquitoes cannot get this olfaction, cannot recognize human, then we do not have any problem, right. Right. So, now they try to understand whether this mosquito's antenna contains some of the olfactory receptors. Okay, so, they take the electrophysiology experiments, they try to see the olfactory receptors in the antenna, they found most of this, they have these receptors, this is how they recognize the human, right. Also instead of the morning time, if you compare the evening time, it can easily recognize human, this way we have lot of mosquito bites in the evening, right. So, now there are several research going on to understand the olfactory receptors mainly in the GPCRs. See, there are several methods they have developed to identify these receptors and see how the mosquitoes recognize this type of receptors and what is the correspondence between the proteins in the mosquitoes and the proteins in the human, and see how what is the correspondence between these two to understand the olfactory receptors and address the issue of malaria. So, now we look into these functions, right. There are various functions we described. Can you remember some of these functions till we discussed till now? Antibodies, structural proteins, enzymes, transporters, regulatory proteins, receptors, transporters, right, channels. So, we discussed various functions, right, and the functions of these proteins mainly depends on the structure, right. Structure means I can say okay, here I can show one of the structures, right. How a structure looks like, this will help to detect the function. For example, I discussed about the enzymes, so the active sites. So, if you look into the structure, active sites are not located inside the structure, right, because inside the structure it is completely compact, right. So, you can see where you can see the probable active sites, so that the ligand molecules can enter and interact or any other molecule can go and interact. So, if you go to the structure, if you see this one, you can see a kind of packet here, right. So, if you have different structures, right, now from these structures we can understand the probable active sites and where the ligand can interact as well as they inhibit the activity, right. Currently we know that we have suffered with various type of new diseases, right, cancer and HIV, and you can see the chikungunya, dengue and all, right. So, all these diseases they are targeted by some of the specific targets, proteins, right. So, when you try to design inhibitors, they like to have the structures because the function is dictated mainly by the structure, right. So, in different levels. So, this as I explained earlier, so they came as a case of the hemoglobin, right, the glutamic acid 6 is mutated to valine, so it causes the sickle cell anemia. Likewise, a small change in the hormones like the leucine to arginine, it causes high blood pressure. Like in the case of several proteins like the insulin, right, some changes cause the diabetes, and like the P53 you can see, right. So, here small changes, right, it causes which disease? Cancer, right. So, if you understand the functions and if you understand the cause for the different diseases, it is important to know about the structure, right. So, if I say one example, okay, this is the P53, right. Here you can see a protein is here and you can see DNA here, this is DNA, here you can see a protein, okay, this is a DNA binding protein, right. So, the mutations in the P53, so if you make small changes, mainly the replacement of arginines, right, it causes human cancer. So, now how to understand why these specific mutations cause cancer, what is the relationship between this function as well as from the structure, right. So, in this case you need to get the structural information because the structures are known. So, you can see the proteins, right, this is DNA and you can see the sites where the proteins can interact. Okay, we see the DNA side, you can see here the region where the proteins can interact with the DNA, right. So, when you mutate these amino acid residues which are binding with the DNA and see how their activity changes or how they decrease or increase the binding free energy or the binding specificity. So, whether you see the DNA binding with the proteins, and this will help you to understand how the specific mutation or the structure is important to understand the binding specificity of these protein DNA binding complexes, as well as how this will affect to the disease, for example in the case of cancer. So, likewise they try to get the features, they try to understand the molecular mechanism or the basis of the diseases based on the structures. So, to understand the function it is essential to know about the structures, and this is the reason why it is much emphasis is given to understand different structures. So, when you look into the protein structures, right, so there are various levels of protein structures, right. So, primary structure, secondary structure, tertiary structure and the quaternary structure. Just I will mention these names and we will discuss the details in the next class, right. So, what is the primary structure? Primary structure deals with the combination of amino acid residues. So, we can get the combination of amino acid, okay, first residue is methionine, second residue is isoleucine, third residue is lysine, right. So, you can get this information and go to next level, right. Here you can see the arrangement of these residues, right. Here this is the just the amino acid sequence, right, and when how these residues are arranged, right. So, this will give you a next level of information. So, this is why they called as secondary structure. So, here if you see they are kind of a helical shape like a spiral shape. So, this is why they call this as helix, right, and then the second one you can see there is the kind of a ladder, right. So, this is a kind of a ladder. So, in this case they put this a strand. So, first letter is Greek is alpha. So, they mentioned helix as alpha helix and the second letter is beta. So, they put beta sheet, right. This is secondary level and then go to tertiary level. So, if you see the secondary level and tertiary level you can get more information, right. Here exactly how your protein looks like, how the residues or the atoms are arranged in a 3D structure. So, you get the complete picture of all the atoms when in the case of the coordinates like x, y, z coordinates, right. This will give you a tertiary structure. Then we go one step further. So, several tertiary structures they form subunits, they collectively form the quaternary structures, right. So, if we say this is one chain, this is another chain, right. The biologically they perform the functions once they form these oligomers. If it is one it is monomer, if it is more than one chain we call it as oligomer, right. So, here you can see the two molecules, right, they are called a dimer, right. So, they are very important for the function, right. So, this type of structure this assembled subunits they are called quaternary structures, right. So, in the next class we will discuss more about the primary structure, secondary structure, tertiary structure and quaternary structures and what are the information we gain from the primary structure or secondary structure or tertiary structure and how this information will be transferred to understand the mechanism as well as to understand the function. Fine. Thanks for your attention.